The global rise of antibiotic-resistant bacteria, particularly Staphylococcus aureus, poses a critical threat to public health. Traditional antibiotics are losing their efficacy, leading to prolonged illnesses, increased healthcare costs, and higher mortality rates. This necessitates the exploration of novel, cost-effective, and readily available antibacterial alternatives. In line with the Harmonized National Research and Development Agenda of the DOST for the year 2022 to 2028, this study is proposing a development of a possible antibacterial with the use of the plant, namely Portulaca Oleracea or Ula semen. In related studies, the Oleracea extract contains alpha-linolenic acid, flavonoids, particularly cankerol and epigenin, saponins and tannins, which made the researchers think that it could possibly possess antibacterial against S. aureus. The primary aim of this study was to investigate the antibacterial effect of Purslane plant Portulaca Oleracea crude extract against Staphylococcus aureus. This research was based on the fundamental concept of antibacterial activity, which refers to the ability of a particular substance or compound to inhibit or prevent the growth and proliferation of bacteria. In this study, the researchers utilized a true experimental research design adapted from the studies of Baserate et al. 2015 and Debalke et al. 2018 to test if the crude extract of P. oleracea possessed antibacterial activity against S. aureus. In the experimentation, the researchers prepared 5 treatments for each plant parts with 3 varying concentrations of P. oleracea leaves, stem, and roots, and negative and positive control, having a total of 15 varying treatments used in the study, while employing the 3 trial replication in every treatment to establish the reliability and consistency of the result. This design was also used to determine the most effective plant part extract in terms of bacterial inhibition. Prior to the conduct of the study, plant samples of P. oleracea were first identified and verified by DENR Penrock at Calogan City. Then the researchers coordinated with the RIC Microbiology Laboratory to obtain S. aureus, from which this culture was taken from DOST Regional Standard and Testing Laboratory LES 2021 by the university, then stacked and cultured in the laboratory. To ensure a representative sample, Purslane plant components were collected from multiple locations within Catbalogan City proper, specifically Mabini Avenue, San Roque Street, and Barangay 8 Poblacion. Purslane plants were randomly selected from each location and then divided into leaves, stems, and roots to achieve a balanced morphological representation. A total of 150 grams of each plant parts were collected and weighed using an analytical scale. These samples were then extracted through 48 hours maceration in ethanol, and the resulting filtered extract was concentrated using a rotary vacuum evaporator. Immediately following extraction, the crude extracts from each plant parts underwent phytochemical screening to confirm their antibacterial properties. Nutrient agar was used as the medium of this diffusion assay. The DOST diffusion method was used in the study to determine the growth of inhibition of bacteria in plant extract as described by the Kirby-Bauer diffusion method. The filter paper discs were impregnated with the extract and were placed on the nutrient agar media with a distance of 24 millimeters of each disc. The growth of inhibition was recorded after 18 hours of incubation. In determining significant differences in antimicrobial resistance pattern of S. aureus among the different treatments and among plant parts, two-way ANOVA was used. And in determining the most effective specific plant part and concentrations between treatment groups, Tokay-Kramer post-hook test was employed. Based on the results, all the treatments excluding T0 and T1 showed zones of inhibition in S. aureus bacteria, and the concentrations of P. oleracea crude leaf, stem, and root extract that is most effective is treatment 3. Moreover, among the 3 crude extracts at their most effective concentrations, statistical findings revealed no significant differences among the plant parts. Furthermore, results also indicated significant differences between the most effective concentration of P. oleracea leaf, stem, and root and the positive control gentamicin. Therefore, P. oleracea crude extract is an effective antibacterial against S. aureus. These findings is significant to the wider community by offering a potential accessible and cost-effective natural remedy against antibiotic-resistant Staphylococcus aureus, reducing reliance on expensive pharmaceuticals. This could lead to improved public health outcomes, especially in resource-limited areas, and promote the use of locally available resources for medicinal purposes. Additionally, it provides a scientific basis for further exploration of Purslane's therapeutic potential leading to the development of new natural antibacterial treatments.